Have you ever heard of cystic fibrosis? Did you know it's considered a cell membrane disease? Cystic fibrosis, or CF, is a genetic condition that affects over 160,000 people worldwide. It mainly affects the lungs and digestive system. CF causes thick, sticky mucus that makes it hard to breathe and difficult to digest food. But where does this all start? It begins with a gene, a segment of DNA that contains the instructions for making a specific protein or functional molecule in the body. In people with CF, there's a mutation in a gene called CFTR. This gene carries instructions for making the CFTR protein. If we zoom into the lung region and then into the thin lining of the airways, we'll find CFTR proteins situated on the surface of epithelial cells. These proteins form a specialized type of chloride channel that sits within the cell membrane. This channel controls the flow of chloride ions in and out of cells. Why is that important? Because where ions go, water follows. This creates a thin layer of mucus that coats the airway, allowing it to trap dust and microbes. Hair-like projections called cilia then push this mucus, along with any trapped particles, up and out of the lungs, helping to keep the airways clear. But in cystic fibrosis, CFTR protein is faulty. It might be misshaped, missing, or just not working right. In a CF cell, the CFTR channel is unable to transfer chloride ions out of the cell and onto the airway surface. As a result, water doesn't follow the chloride ions out of the cell by osmosis, leading to the buildup of thick, sticky mucus on the airway surface. This mucus clogs the airways, making it difficult to breathe and increasing the risk of infections. That's why people with CF have trouble clearing mucus from their lungs, and why they're more prone to lung infections. And this is just one single example. Cell membranes are fundamental for all known life. Seriously, every living cell has one. They are the ultimate gatekeepers of our cells, the bouncers deciding who gets in and who stays out. So let's dive deeper into how these amazing structures work. Imagine a cell as a super busy tiny city, full of workers, factories, and power plants. The cell membrane? Think of it as border control and city walls all rolled into one. Let's take a closer look at the cell membrane. It might look complicated, but it's not static. In fact, it's fluid and flexible, often described as a fluid mosaic. The molecules within it are constantly moving, allowing the membrane to adapt and respond to its environment. The majority of the cell membrane is made up of a double layer of phospholipids, also called the lipid bilayer. Think of phospholipids as tiny balloons with two strings. The balloon part, the head, absolutely loves water. We call that hydrophilic, which means water-loving. The two stringy tails, though, they despise water. The term for that is hydrophobic, which means water-heating. This love-hate relationship with water is key. It dictates how phospholipids organize themselves. The water-hating tails, made of long, non-polar fatty acid chains, avoid water because they don't mix well with the polar water molecules. As a result, these hydrophobic tails face inward, clustering together like the filling of a molecular sandwich. Meanwhile, the water-loving heads contain a polar phosphate group that interacts strongly with water. These hydrophilic heads face outward toward the watery environment outside the cell and the watery cytoplasm inside. This unique arrangement forms a flexible barrier that protects the cell while allowing it to interact with its surroundings. But wait, cell membranes aren't made of just lipids. Proteins float in the lipid bilayer like icebergs in a sea. Some, like CFTR, act as channels, while others bind to carbohydrates and help with cell recognition and communication. There are three main types of transport mechanisms across the cell membrane: passive transport, active transport, and bulk transport. Passive transport is the simplest type of cell transport. It relies on molecules moving from an area of high concentration to low concentration and does not require energy. There are three main forms of passive transport. Simple diffusion, where small molecules like oxygen move directly through the membrane due to a higher concentration outside the cell. For larger or charged molecules, such as glucose or ions, facilitated diffusion occurs, requiring protein channels to assist their passage. These molecules cannot pass directly through the lipid bilayer due to their size or polarity, so the channels help them move down their concentration gradient without using energy. Osmosis is a type of diffusion that involves only water. It occurs when water moves across a membrane from low solute concentration to high solute concentration. This movement balances the solute concentrations on both sides. All of these happen automatically, no energy needed. Active transport requires specific transport proteins and energy. This energy, typically provided by ATP, is used to move molecules against the concentration gradient. When ATP binds to the transport protein, it triggers a conformational change that enables the molecule to be transported across the membrane. One example is protein pumps, such as the sodium-potassium pump, which uses ATP to move ions against their gradient. However, some molecules are simply too large to pass through by diffusion or pumps. This is where bulk transport comes in. Bulk transport moves large substances or large quantities using vesicles and also requires energy. There are two types. Endocytosis is when the cell membrane folds inward to take in substances. To help you remember, the N stands for enter. This process includes phagocytosis, cell eating, in which the cell engulfs and internalizes solid particles, such as bacteria or cellular debris, into a vesicle for processing or digestion. Pinocytosis, often called cell drinking, is a type of endocytosis in which the cell engulfs small droplets of extracellular fluid. The opposite process is called exocytosis. This is when tiny vesicles inside the cell move toward the membrane, fuse with it, and release their contents outside the cell. For example, this is how your nerve cells release neurotransmitters to send signals to the next cell, or how hormones like insulin get released into your bloodstream to regulate blood sugar. And here's an easy way to remember it. Exocytosis means exit. It's the cell's way of sending things out. So far, we've seen that the cell membrane isn't just a barrier, it's a dynamic and highly organized system. Diseases like cystic fibrosis show what happens when membrane transport goes wrong, while the interplay of lipids and proteins controls how substances move in and out through diffusion, active transport, endocytosis, and exocytosis. Understanding these processes drives exciting areas of current research. For example, scientists are developing targeted cancer therapies that deliver drugs directly into tumor cells by exploiting endocytosis pathways using liposomes, which are tiny lipid-based vesicles, similar to the phospholipids that make up our cell membranes. Liposomes are tiny, spherical vesicles made of lipid bilayers that can encapsulate chemotherapy drugs. As they circulate through the bloodstream, they can be engineered to recognize and bind to specific receptors on cancer cells, ensuring the treatment is delivered precisely where it is needed. Once bound to receptors on the surface of cancer cells, liposomes are internalized through the endocytosis pathway, where they gradually and precisely release the drug directly inside the tumor cell. This targeted delivery enhances treatment effectiveness exactly where it is mostly needed, killing cancer cells while minimizing harm to healthy cells. Even though the cell membrane is tiny, it plays a central role in some of the most promising breakthroughs in medicine and biotechnology. Understanding it today is paving the way for the innovations of tomorrow.